neat features to it. Certain bases pair together, and the I got distracted. Hello and welcome to biology. You've been learning a lot about DNA recently, and DNA you learned has two strands. It has different bases that pair together, A's with T's and C's with G's, and you may have even learned that it's the order of the bases, the order of the A, T, T, C, A, G, C, whatever the order might be, that determines your traits. And while that's all true, in the end, your traits are the result of the proteins that DNA tells your body how to make. In the picture on your left, you're taking a look at a portion of a DNA sequence, and we call those portions genes. And in the end, it's just a code. The code is great, but in the end, it allows your body to make a protein that does some sort of work for you. And so, for example, you have an eye color gene, maybe blue eyes or brown eyes. And in the end, you don't get to see ATTCAGC. You don't get to see the DNA sequence. You see the brown eye or blue eye protein that's produced. You're able to carry oxygen in your blood because of a protein called hemoglobin, shown here on the right, not because you have a DNA sequence of a certain set of bases. Those bases, in a particular order, are a code to tell your body how to make a final product. And so we need to spend some time figuring out what that final product is. What are proteins? What do they do for the body? How are they put together? And at some point, take a look at the mechanics of how information in DNA is used to build proteins. In some regards, it's kind of like somebody who's studying to build a house or learning how to build a house, like an architect. And an architect can sit there and make blueprints all day, and those blueprints are great. They're very important for knowing how to build the house. But if all the architect does is build blueprints, then in the end you don't have a final product. You can't stand in certain parts of the blueprint and admire the kitchen. All you can do is look at the blueprint and envision what might actually be produced. In the end, it's the final product that we're concerned about. We really, in the end, are having that architect work to build a house. So proteins are important. Here's a quote here that I think is relevant, that when the body has something it needs to do, it's a protein that does it. So let's give you some examples of that. Examples of how DNA is a blueprint, but in the end it's the proteins that do the work. Some of these proteins you've already been learning about. Back when we learned about the cell membrane, we learned about the phospholipid bilayer, but we also learned that there are proteins like this receptor here, or channels even, that open up and allow certain molecules very specifically to move into or out of the cell. Certain proteins, like receptors and channels, control what enters and leaves the cell. Other proteins include checkpoint proteins that we learned about during the cell cycle, that during the process of going from interphase through telophase and then back again, the cell needs to proofread and make sure that it's doing those steps correctly. And that's what checkpoint proteins do. And as I showed you on the first slide, hemoglobin would be one example of a protein that transports things. In particular, hemoglobin is found in RBCs, or abbreviation for red blood cells. And it's this hemoglobin protein that is able to carry oxygen for you. Without hemoglobin, there is no way to carry oxygen, at least safely, from where you breathe it in your lungs to the cells that need it. And so that hemoglobin protein is important, but there are a wide variety of other transport proteins that help to carry other molecules as well. Antibodies. When you get sick, your body produces antibodies in ways that we'll learn about next semester, and these antibodies help to fight diseases. Each protein, each antibody is able to fight off a different disease. You just have to be able to produce the right ones. Enzymes play a key role in your body's ability to stay healthy. Enzymes are proteins that act as cellular machines. For example, you recently learned about DNA polymerase, shown in the background in this image here. DNA polymerase able to look at both strands of DNA and make a copy of that DNA sequence. Other enzymes are found in your stomach to help you digest your food. Other enzymes are found in a whole bunch of other examples of cells because in the end your body is doing thousands and thousands of chemical reactions every single second, and virtually every single one of those reactions requires an enzyme in order for those chemical reactions to work correctly and quickly. So enzymes, another example of proteins. Hormones. Some hormones are proteins. They tell the cell what to do and when. Back in cell unit, we learned about insulin as a signal to tell the body that sugar is present, and that insulin was able to bind to an insulin receptor and ultimately tell a glucose channel to open up. Without insulin, without that signal, the body wouldn't know what to do and when. Hormones, some hormones are examples of proteins too. And what I will add in here too, movement. Movement, in order to move, your muscles are able to contract as a result of proteins called actin and myosin. And even structural proteins, ones that seem to just be part of your body and don't really do anything active, are still an important example of proteins. Fingernails, for example, are made out of proteins called keratin. Hair also. So a wide variety of proteins that your body uses. And each one of these proteins has its own unique shape. Antibodies look a certain way, and you can go back here and show you what they look like. In general, they have this sort of Y shape. Hemoglobin has a very specific shape there. We can take a look at other proteins like receptors and channels and DNA polymerase, and every single protein, these hormones, every single protein has their own unique shape, even within antibodies. An antibody to fight off a common cold from this past year, even versus the common cold from 3-4 months ago. That virus has changed. And as a result, you have a different antibody with a slightly different, I should say, shape to be able to fight off that disease. In order for that protein to work, it has to have the right shape. What determines the shape of a protein are its building blocks. Remember, DNA was made out of nucleotides, a sugar, a phosphate, and one of 4 possible bases. For proteins, amino acids are the building blocks of proteins. And I want you to make this sketch into your notes here. You can go ahead and pause the video if you need to. But I want you to make sure that you have a carbon here in the middle. That's the C. That you have a hydrogen. And then off on the side here you have this HNH. This is an amino group, hydrogens and nitrogen. Off on the right you have carbons and oxygens and hydrogen to make an acid group. Don't worry so much about the number of these lines here. These are bonds that form, and at some point in chemistry you'll learn how many bonds an oxygen can have and how many bonds a hydrogen can have and so forth. But this is the basic structure here. This top part is the basic structure for all amino acids, and all amino acids have this extra piece hanging off the end of it too that we call a side chain. There is no element in the periodic table with the letter symbol R, but what R represents is one of 20 different possible side chains. And I'll show you examples of those side chains in a moment. So if you haven't paused it yet and labeled this diagram, please do. Otherwise, we're moving on. 20 different amino acids that the human body uses, each one of them with their own R group. The R group is shown in pink. The rest of that basic amino acid structure, to go back for a second, the rest of it, the amino group, the carbon, the hydrogen, the acid group, the rest of that is shown here, but each of these 20 different amino acids has their own unique side chain, or R group. And some of these are very short. You'll see here that glycine has just simply hydrogen. Even alanine is relatively simple in comparison to things like glutamine down here that have a much longer chain, or histidine, which seems to be curved in on itself, or arginine, which is really long. Each R group gives each amino acid, I should say, its own unique characteristics. Some are short R groups, and so they vary based on size. Some are large amino acids and some are small amino acids. Some amino acids are differently shaped based on their R groups. Some amino acids have a positive charge, some amino acids have a negative charge. These up here, lysine, arginine, histidine, all have a positive charge, whereas these, aspartate and glutamate, or asparagine or glutamine, are negatively charged. And some have a preference for water, and some hate water. Those that hate water are hydrophobic, and those amino acids that have an R group that prefer to be in contact with water are hydrophilic. And we learned those terms back in the cell membrane portion of this semester. Different amino acids give each amino acid, or sorry, different R groups give each amino acid their own unique characteristics. In order to make a protein, it's not just one amino acid though. We're going to take one amino acid, one of those 20, and connect it to another one of those 20, and keep going one after the next after the next. These amino acids are connected by special bonds called peptide bonds. Here in this example, we're seeing one amino acid off on the left. There's its R group. They drew it up top instead of down below. That's okay. And here's another amino acid with its R group. And through a reaction that we'll learn about next semester, we can lose water to connect these two. An amino acid on the left, a second amino acid on the right, and this green line here is a peptide bond to hold those two amino acids together. Proteins are more than just two amino acids linked together. They are hundreds and sometimes thousands of amino acids connected, and so each amino acid linked into that chain requires another peptide bond to connect them. So I want you to make sure that you circle in that diagram an example of where that peptide bond is. In order to make a protein, then, the first step, this primary level of structure, is simply just the order of the amino acids. Which beads are you going to place into that chain first? And so these circles represent amino acids, the lines would represent peptide bonds, and this primary structure of protein shape is based on the order and type of amino acids that are used. And then proteins do something different. It's not just the order. What happens next is that they start to fold in on themselves. A level of structure that's referred to as secondary, that some areas of the protein link, the chain of amino acids, are going to coil in on each other. We call that an alpha helix. And in other cases, they kind of fold together like an accordion or a beta sheet. So to go back a picture here, some amino acids love to be in contact with each other. So maybe these two love to be in contact with these two here, and so they're going to start to link together and form a connection. And maybe even these two with these two start to form a connection. They don't have to be physically right next to each other, at least in order in the chain. They can fold up in a whole bunch of different arrangements. And there are researchers now who are busy trying to make predictions about, given a length of amino acids, how that protein might fold into a particular shape. After they start to coil and fold together, they fold and coil even a little bit more. We call that tertiary structure, how all the helices and sheets fold together. And there's even a fourth level, a quaternary structure, where some proteins are made out of more than one piece, more than one subunit. So one piece of that protein is going to have to fold in one area of the cell, another one in another area, and then they're going to have to come together. An example of this would be something like hemoglobin, that is made out of two different subunits that have to come together. In the end, actually, four. And the point here isn't to really kind of have you memorize what primary, secondary, tertiary, and quaternary structure are, but is really just to point out that the shape of this protein ends up being very different from one protein to another, and is a big part of how that protein works. That in the end, how that protein folds is determined by the type and number of amino acids that are used. In order to get a shape like this, you have to have the right amino acids in the right order within that chain. Otherwise, you'd never be able to fold that up. It'd be like trying to fold a piece of paper into a paper airplane. But if you aren't using the right type of paper, or if you aren't using the right folds, then you aren't going to be able to make the shape that you want, and that plane isn't going to fly. The same is true here for proteins and how they fold. And of course the instructions to be able to tell a cell which amino acid to use first and second and third. The instructions to tell the cell which amino acid to use is the DNA sequence, the order of those A's, T's, C's, and G's. So DNA sequence is important, but in the end, you have to have the protein that's doing the work. So how does the body know how to make proteins? Well, it's, again, based on the DNA sequence. If the order of those nucleotides is changed, then the sequence of amino acids is changed, the shape of that protein is different, and that protein is going to function differently and most likely isn't going to do the work that you want it to do. To highlight this point, we're going to be watching a video from the movie that we saw recently called Cracking the Code. It's Chapter 9. I've given you a link at the website in the video section, and I do want you to watch this video. It's going to show you how these proteins here in the cell membrane, so the phospholipid bilayer, this is a salt channel, and at some point this channel needs to open up to allow salt, here these little chloride ions, it needs to allow salt to move from one location of the cell to the next, from inside the cell to outside. But there are big problems that can happen if that protein, this salt channel, can't do its work. So I'd like you to watch that. If you're watching this outside of class, inside of class, we'll probably take a chance either today or real soon to watch that together. And certainly if you have any questions, please let me know.